Hello, I'm Frank Delaglio from NIST. I'd like to thank LabRoots for this opportunity to share the work of our labs. Today we're going to talk about characterization of biotherapeutics by NMR and about the analytical methods and computational methods that we use to extract useful information from these spectra. And the work that we're going to be presenting today has been conducted at NIST by many collaborators, including the folks in my group, Luke Arbogast, Robert Brinson, and our group leader, John Marino, postdoc Korth Wade Elliott, and on the NIST main campus, our computational colleagues, Ryan Evans, Tony Kearsley, Felix Perez, and Christopher Schanzel, and the ITL, our colleagues, David Sheen and Vincent Shen. What's our motivation for studying biotherapeutics? Well, if we look at a list of the top 10 drugs by sales, what we discover is that 7 of these are biologics, that is to say, protein drugs. And of those, 6 of them are monoclonal antibodies, what we also call MABs for short. There's 2 stories in this table. So one is, of course, that these protein-based drugs are important, life-changing, life-saving therapeutics, which is why they're at the top list here. But in addition, they're also incredibly expensive, with some treatments costing as much as $100,000 or $200,000 for a year's worth of therapy. So any analytical and computational methods that we can bring to bear to make this important class of therapeutics more accessible, more affordable, more stable, easier to develop, those can have impact. And so that's what we're going to talk about today. As a little digression, it's useful to consider what traditional drug discovery has been about. Small molecule drug discovery, like aspirin, Tylenol, and so on. So most small molecule drugs work by targeting a specific protein or class of proteins, like the one shown on this slide, which is cyclooxygenase, or COX. This protein catalyzes creation of prostaglandins that promote inflammation and thromboxanes that promote blood clotting. So if you can find a small molecule that will interfere with this protein, you can prevent blood clotting and prevent inflammation responses. And that's how meds like aspirin work. Now, it was known for more than 2,000 years that willow bark, which contains salicylic acid, was a good way to reduce fever and inflammation. And based on that understanding over time, when people deduced the structure of salicylic acid and had the wherewithal to make chemical modifications, drug companies came up with the modification aspirin, and then later on, additional systematic modifications like acetaminophen and ibuprofen. And so this has been a kind of paradigm for small molecule drug discovery. We have buildings full of synthetic organic chemists making small chemical changes to common molecular scaffolds and testing, generally by trial and error, whether or not a particular molecule can bind to the target protein of interest. And in this case, with the cyclooxygenase, many different small molecule drugs have been found to bind to this protein and make a useful drug. There's a kind of corollary to this, however, which is that if you can find many different small molecules that will all work to target this particular protein, it also implies that these small molecules might target other proteins that you don't really want targeted. In other words, when you make a small molecule drug, it will commonly have side effects. And in fact, this is often why drugs fail. And the failure rate is about 90%. And in spite of all the additional information we brought to bear about structural biology, that failure rate hasn't changed much. So small molecule drug discovery still depends on trying large numbers of compounds against a drug target. And a big pharma company might have compounds, several million compounds in a library that they can rely on. So that's small molecule drug discovery. Of course, proteins themselves can be used as drugs. This is what we call biologic therapeutics or biologics. And probably the most well-known and common one is insulin. That regulates glucose levels. And there are other ones that we know about, like epogen, filgrastim, that stimulate production of blood cells. So these are all naturally occurring proteins that we can manufacture in cells, in living cells, extract them, and administer them as drugs. Now, we notice that there are many monoclonal antibody drugs in the list of the top 10. Why is that? Well, we already considered that in small molecule drug discovery, trying large numbers of different compounds against the target is a good way to find particular compounds that will hit your target of interest. In the case of antibodies, these have been designed by nature to vary in such a way that allows them to bind to many different targets, very specifically. So here's a little cartoon of what an antibody looks like. It has 3 domains. The little donut-shaped domain at the bottom is what we call the Fc domain. This part binds to the immune cell receptor. And at the top are the 2 identical Fab domains of the antibody. And at the ends of the Fab domains are the places that bind to the antigen, whatever it is that the monoclonal antibody is targeting. Now, we talked about the fact that a big drug company might have 5 or 10 million compounds in their small molecule database, but an individual human can make over 10 billion different kinds of antibodies. So antibodies are an amazing source of structural diversity, and bioengineering allows us to select particular antibodies that will bind very specifically to pretty much most any target that we like. And so for this reason, and for some others, monoclonal antibodies have become very attractive vehicles for drug discovery. Recognizing that at NIST, where we like to help the development of measurements and standards, we developed a standard monoclonal antibody reference that can be used to develop and validate technologies for studying therapeutic proteins like monoclonal antibodies. And we're going to use this reference antibody called the NISTmAb for several examples in the following slides. Now, in the case of a small molecule drug like aspirin, it's an organic molecule, and we can specify pretty much everything about it according to its network of chemical bonds, its covalent structure. But the situation for protein therapeutics is different than that. The covalent structure, what we call the primary sequence of the protein, the amino acid sequence, can be determined very accurately by techniques like mass spectroscopy. However, in order for the protein drug to function properly, it has to be folded into a very specific structure, and it has to stay in that structure and not aggregate. So in the case of protein structure, or what we may also call high order structure, there's a hierarchy of organization, starting with the primary sequence, the secondary structure, small structural elements like helices and sheets, the overall fold of the protein, and then finally any complexes that the protein might form. In order for the protein to be safe and efficacious, it has to be folded properly, and it can't be aggregated. Because if it's aggregated, even in such a way that preserves its activity, it can present a different shape to the immune system, an unfamiliar shape that will cause an immune response. The immune response might make that dosage of the medicine ineffective, or it might threaten the life of the patient. So, important challenge, and we have to address it. The difficulty is, as we get higher up this chain, this hierarchy of structure, it becomes more difficult to measure the details of structure at atom level resolution. So we're going to try to resort to the analytical methods of NMR to help us discover the details about high order structure. Now, in order to best conserve the structure and stabilize it from unfolding and aggregation, it's common for these protein drugs, which are administered by injection, to have one or more excipients added to the injection solution to keep the protein stable and unaggregated. And actually deciding which collection and concentration of these excipient compounds are in a particular therapeutic is actually an important aspect of development. So having analytical techniques to assist this would also be useful. Now, in the area of structural biology, where we use NMR, it's very well known that the 2D hydrogen-nitrogen correlation spectrum is closely associated with the particular structure of a protein and very indicative of small structural features. For example, what you see here are two NMR spectra of the protein therapeutic called filgrastim, or GCSF. On the right-hand side is the NMR spectrum of the intact protein properly folded, and on the left-hand side is a spectrum of that same molecule, but in its unfolded state. So this is a rather extreme demonstration. The little spots in the spectrum are like mountain peaks, and the position of one of those spots in the horizontal direction is the NMR chemical shift, the frequency of a particular amide hydrogen atom of one amino acid in the protein. And that peak's position in the vertical axis is the chemical shift of the corresponding nitrogen that hydrogen atom is bound to. So together, the hydrogen and nitrogen are chemically active, and they can be excellent reporters on structural and environmental changes of the protein. And if you have happened to conduct the assignment procedure of mapping out which of these peaks in the spectrum belongs to which amino acid in the protein, you can map changes in these spectra onto the surface of the protein of interest and decide, as in this case, where the spectrum exhibits signs of oxidation of particular methionines, we can see which other parts of the protein get manipulated as well. So NMR gives us a way to extract structural information of proteins in solution, more or less as formulated. But we'd like to apply them to really big molecular systems like monoclonal antibodies, which are several times larger than the proteins usually studied by NMR spectroscopy, which is generally limited by how quickly a molecule can tumble. That is to say, more slowly tumbling molecules make NMR measurements more difficult. And so while a monoclonal antibody is very big, 150 kilodaltons, and tumbles very slowly, the methyl groups of side chains in a monoclonal antibody are reorienting very rapidly. So even though the protein as a whole is tumbling slowly, we can measure spectra very effectively without isotopic labeling for the carbons and protons of methyl groups in the protein side chains. And about a third of the residues in a protein have such side chains. So this is an excellent way, what you might consider a fingerprint of the protein structure. And if the protein structure changes in some way, either chemically or structurally, we'll observe changes in the NMR spectrum. So what I'd like to do is show you what kind of changes we can induce in the protein as a test case and how we can detect these changes by NMR spectroscopy. Now, monoclonal antibodies have sugar groups attached to them, glycans, and these sugar groups strongly modulate how the antibody can interact with an immune cell receptor. So the composition of these glycans is incredibly important, and small changes in manufacturing conditions can change the profile of glycans that are attached to the antibody. In the case of our NISTmAb reference monoclonal antibody, the majority of glycan sugar groups fall into these three categories that are shown here, called G2F, and they're called such because the two ends of the glycan end in the sugar group galactose, two of them, or G1F, where only one of the two termini has a galactose sugar, or G0F, where there are no galactoses at the end. Now, as mentioned, the presence of these glycans, these sugar groups, can strongly change how an antibody binds to an immune cell receptor. And that's indicated in this cartoon of the Fc domain, the donut shape at the bottom, being bound to an immune cell receptor protein in green at the top. So we know that the glycans change the nature of this binding. But interestingly, when we look at X-ray crystal structures of this complex, what we see is that the glycans don't come into direct contact with the immune cell receptor. So the way that they change binding with the immune cell receptor is by slightly changing the structure and dynamics of the parts of the monoclonal antibody that come into contact with the receptor. So we can use the glycans as a way to make subtle structural modifications to the protein and detect whether or not we can see such changes and characterize them by NMR. We have a whole host of enzymatic processes that we can use to modify and remodel the glycans on our antibody. We can use enzymes that will add additional sugars, as we see on the top left. We can use enzymes that will strip the terminal sugars, like at the bottom on the left, or we can use enzymes that will clip the entire glycan group off completely. So these are the test cases that we're going to look at. Here's a collection of 34 different NMR spectra that were measured with different parameters on 3 different kinds of samples that we made from the NISTmAb. One sample is the native version of the NISTmAb. The other is a version that's called G0F. We used an enzyme to clip off the 2 galactose residues. Or in a third sample, we used an enzyme that trims off the glycans completely. So a deglycosylated sample. So 3 samples, 34 spectra measured under different conditions. I have to say that a difference between the native NISTmAb and the G0F glycoform is a matter of changing only about 3 sugar groups in a molecule with 1,400 amino acids. So it's a very minor change, and it will be interesting for us to see that we can pick it up with NMR. Now, when you look at these spectra, you can see that a majority of the peaks are in a central blob, and you can imagine that that would be very difficult to analyze by eye, and if you imagine that, you'd be exactly correct. So we'd like to use some computational methods that will assist us in analyzing this spectrum. And we're going to use methods that will work directly on the matrix of spectral intensities, so that we'll make our analysis based on the shapes of the spectrum rather than any detailed interactive analysis of individual peaks in the spectrum. And the method that we're going to use is principal component analysis, or PCA. And in this application of PCA, we will use the spectra directly. And we're going to start by using the collection of spectra in a multivariate or multidimensional representation, where each spectrum in the series to be analyzed is represented as a single object in a big multidimensional space, and the coordinates of that object are simply all of the intensities in the spectrum. This means we can use the spectra directly without any need for peak analysis. Now, what does that mean specifically? Well, let's say we have a 2D spectrum that's 1,000 by 1,000 points. That means 1,000,000 total intensities in the intensity matrix. We can represent such a spectrum exactly as a single object in a 1,000,000-dimensional space. Now, this seems peculiar, but if you think about this data representation, you'll see that it has some interesting and useful properties. For example, if we think about two spectra which are more or less the same, that means their intensities are more or less the same, and that means that they correspond to objects that lie in more or less the same part of this large multidimensional space. Whereas if we have two spectra that are mostly similar, but perhaps they vary in one place because a peak moves a little bit from one location to another, spectra like that are going to tend to lie along lines and curves in this large multidimensional space, because they're going to have one set of coordinates in common, that is to say, all the parts of the spectra that are the same, and some small subset that vary continuously, those parts of the spectrum that are different. So this large multidimensional space will group together spectra according to their degree of similarity and difference. And all we need is a way to reduce that large multidimensional space of 1,000,000 dimensions or so down to a small number of dimensions that are reasonable for us to visually inspect or do easier computations on. And the technique that we use is principal component analysis, which simply fits a multidimensional straight line through the data and projects it down onto that line to reduce the dimensionality of the spectrum collection. And so if we take this large dimensional space and project it down, let's say to 2 or 3 dimensions, we should be able to observe clusters and patterns that group together the spectra according to their similarities and differences. There are 2 ways that we can visualize the results. One is that each principal component that we calculate results in a kind of synthetic spectrum that represents that component. So one thing we can do is visualize the component results as spectra. And the other is that we can make scatter plots of the object coordinates of the principal component results. So let's look at some results for this collection of data. If we visualize the principal component results as a spectrum, the first principal component you could think of as a spectrum that best describes the entire series in a least square sense. So this first component is more or less like an average spectrum. It shows us what's in common among all the spectra in the series. And in this case, we haven't changed the protein itself. All we've done is change the glycans. So there's only a small difference in all of those spectra in the series. But now, if we subtract this and look at the second component, what we see is that there are particular locations in the spectra that are changing from one spectrum in the series to the other. So here's the second component, here's the third component, and here's what they look like together. If we have knowledge about the particular assignments, that means we can map this information onto the protein and decide what parts of the protein are being affected by the changes that we've performed. But in addition, in this case, we don't happen to have specific assignments yet, but we do know generally which kind of amino acids the different regions of the spectrum represent. So we can, for example, tell specific changes to methionine residues and so on. But what happens when we look at the principal component results as a scatter plot? It's very nice. What we see is we can get nice clustering according to the sample type. So down below in blue are all the spectra of the native NISTmAb. In the middle are the spectra that are associated with the G0F glycoform. And at the top, spectra of that sample that was partially deglycosylated. So without any interactive analysis, without the need for perfectly resolved peaks, we can go from a result, which is just a stack of spectrum, to a classification result like this. Now, in this case, the colorization and clusters were done manually by people who already knew which sample was which. But now we can bring to bear the techniques of machine learning. So in this case, we're going to apply a method called hierarchical clustering. In this case, this is complete linkage analysis. And in this method, each spectrum starts out in its own cluster, and at each iteration, we join together the two closest clusters into a single group. And we repeat that procedure over and over again until we've achieved the desired number of clusters. So here's what the result looks like if we request the generation of 3 clusters. And here's the result that we get, 4 clusters, and here's the result that we get if we request 3 clusters. So this is one example of a method where we used unsupervised analysis of the spectrum, and it can return a useful result about the nature of the sample. There are other interesting things that we can do in this way. For example, here's principal component analysis results of mixtures of native NISTmAb with the G0F glycoform. So we get a kind of concentration titration curve, and from this we can estimate the fact that our detection limit is about 10% in this case. In this example, we study spectra from 2 different lots of the NISTmAb measured at 2 slightly different temperatures. And what we see here is that the variation in spectra with respect to temperature is much larger than the lot-to-lot variation. So in this case, this is a kind of proof of concept to show how you would use this method to be able to decide on similarity. Here's another interesting aspect. Post-translational modifications like oxidation of methionines can have important clinical effects, including changing how an antibody binds to an immune cell receptor. Here's an example of 3 kinds of samples: native, one where peroxide was added to oxidize all of the methionines, and a third sample which was prepared in such a way that it performed targeted oxidation of only a subset of all the methionines. Here's what the methionine region of those 3 targets looks like, and here's what the principal component results look like. So without any interaction, we're able to differentiate these 3 cases of native, partially oxidized, and fully oxidized. Another study that we can do by NMR is to measure a degree of molecular interaction. The curve that you see on the left-hand side is an intensity curve with respect to increasing concentration of our monoclonal antibody. And we would expect in an NMR spectrum that as concentration gets higher and higher, the intensity of the peaks gets proportionally higher. But what we can see, somewhere between a concentration of 40 and 60 mg per mil, that linear relationship changes to a different one. What this implies is at a certain concentration, there's some kind of intermolecular interaction that's happening that's changing the spectrum. We can characterize this by principal component analysis. So here's a principal component analysis spectrum. The gray spectrum is the first component, the average spectrum, and the blue and red parts are the higher components. We can see that at higher concentrations, only specific parts of the spectrum are changing. So there are molecular interactions, and they're specific. They're not just due to changes in the overall viscosity of the sample. They're actually particular interactions going on. And we can differentiate, for example, between changes in parts of the spectrum that belong to the Fab domain of the monoclonal antibody, changes in the parts of the spectrum that belong to the Fc domain of the monoclonal antibody, and there are even some signals from the glycan that we can see changed as well. So we have NMR used to tell us that a specific kind of molecular interaction or interaction is going on. The principal component analysis results, when viewed as a scatter plot, also give us a kind of calibration curve of this molecular interaction. So interestingly, if we were doing something like formulation screening and we prepared a formulation that we think should reduce molecular interaction and the propensity to aggregate, we would see where such a spectrum falls on a calibration curve like this and decide whether or not the excipient seems to make things better or worse from an interaction point of view. So those are some of the things that we can use our principal component analysis and NMR for. Now I'm going to turn our attention to a recently completed study called the NISTmAb International NMR Interlaboratory Study. This was a study, we believe the largest of its kind, conducted by 26 organizations in 9 different companies on a variety of NMR instruments. And the organizations that participated were nicely distributed between pharmaceutical companies, universities, and governmental institutions and regulatory labs. In this study, where we had participants from all of these organizations, each participant was sent 2 different NISTmAb samples. And the results of the study have been published in the journal MABS, so you can read about them in detail. As mentioned, there were 2 different samples. One is the Fab domain of the NISTmAb that was prepared by enzymatic cleavage of the NISTmAb sample. And the other was a sample called the system suitability sample. That's a version of the NISTmAb Fab sequence that was expressed in yeast. And originally it was intended that these 2 samples would have identical sequence, but one would be isotopically labeled. But it turned out that as a result of the particular methods we used to express the system suitability sample, it happened to have 4 additional amino acids at the end of the 2 chains of the protein. So its sequence differs in just a little bit from the native Fab. And this gave us an additional interesting source of variation that we could test for. The spectra look like this. And of the 375 proton-carbon spectra that were measured, one interesting aspect of the study was that every spectrum was accepted and used as input for further analysis, even in cases where there was some mistake in the acquisition or hardware difficulty. So some subset of these spectra are actually very poor quality, or even under normal circumstances unusable, and would be potentially difficult to analyze by eye. But we can use these same multivariate methods that we've seen before to nicely classify all of these spectra directly according to whether they are system suitability samples or FAB samples, and that works nicely. We can use these hierarchical clustering methods to do this work, and I will show you an example. So here's the hierarchical cluster analysis being applied to the entire collection of 375 spectra. And in this case, we will stop it at 5 clusters. And from these 5 clusters, we can completely characterize the 2 different kinds of samples. As a final topic, I'd like to turn our attention to a new technique introduced by our colleague Luke Arbogast to help study these protein therapeutics in the presence of excipients. This is a method called Sierra NMR. It's a combination of a special pulse sequence for measuring data and a numerical method for treating the data afterward. Now, as mentioned, an important part of biotherapeutic development is determining of formulation, which excipients should be added and in what combination and concentration. However, it's typical that the concentration of excipient in these samples is often much larger than the concentration of the protein therapeutic itself. And as a result, when we want to measure NMR spectra of samples that include excipients, what we find are situations like this one, where the spectrum is dominated by large excipient signals, and these can distort the rest of the spectrum. The Sierra pulse sequence uses a phenomenon called cross-polarization as a way to selectively suppress signals at particular 2-dimensional locations in the spectrum without changing the rest of the spectrum very much. So this is very beautiful, and in practice, it can be used to target these specific regions, and we're going to see an example. So the regions highlighted here, the 4 regions highlighted in white, are excipient signals from the excipient polysorbate. The signal at the left in yellow is the excipient histidine. So first, we're going to use the Sierra pulse sequence filter to try to suppress these 4 polysorbate signals. So this is what they look like before the Sierra pulse sequence is applied. This is what a spectrum looks like when the Sierra pulse sequence is applied. So we've reduced these 4 signals by between 80 and 90% of their original intensity. And so the tails of these signals no longer interfere with the protein parts of the spectrum, which is what we want. But when we looked at the spectrum, we wondered if, now that we've experimentally reduced the size of these signals, if computationally we can do more to suppress them. And so we applied a method called SMILE for spectral modeling that's used elsewhere to reconstruct NMR spectral data, and used it as a way to build a model of the excipient signals, the leftover excipient signals, and subtract them from the spectrum so that we can generate a spectrum, a clean spectrum, that goes from this, where we have the small residual excipient signals, to a spectrum like this, where all the excipient signals have been subtracted. How does it work? And here's the model that we've subtracted to do the suppression. The SMILE technique takes a spectrum and decomposes it as a series of individual peaks, and keeps adding peaks to the model until the spectrum has been completely described down to whatever desired residual. So on the left-hand side, what you see at the very top is what a particular region of the spectrum, in this case the histidine excipient signal, looks like. And then in the little postage stamp plots on the right-hand side, you can see the collective succession of terms that are added to try to model that measured histidine signal. And in the middle on the left, you can see the model as it gets larger and more complicated to better approximate the measured signal. And then at the bottom on the left, you can see the residuals so far. So we've decomposed our signals of interest into collections of smaller signals, and we can take them all and subtract them from the spectrum to generate excipient subtracted results. So in conclusion, I hope you've been convinced that 2D proton carbon NMR spectroscopy is practical to perform at natural abundance on molecules as large as monoclonal antibodies, and we can get a wealth of information that's useful in the development and manufacturing of biopharmaceuticals. Furthermore, we've demonstrated computational methods that make it possible to objectively and automatically analyze such spectra without the need for extensive interactive analysis. And we've shown computational methods and experimental methods that can be combined to allow us to study these biotherapeutic samples exactly as they are formulated for use. So that's an overview of what we can achieve with NMR, and coupled with methods of machine learning and chemometrics. I hope you found it interesting. Thanks very much. I'm looking forward to your questions.